The cell cycle. There are two stages to cell life, the interphase and the end-phase. Interphase is where the growth and replication of DNA, while in end-phase is where the division of cell. Cells spend about 90% of the time in interphase. Interphase is divided into three phases: the G1, the S phase, and the G2. G1, or the first gap, is where the small cell is absorbing nutrients, growing, and doing its job. In S phase, cell is continuing to give and duplicates, while in second gap, or the G2, is where the cell keep going and doing its job. Then the M phase. M phase, there are two types of M phase, the mitosis and the meiosis. First will be the mitosis. Mitosis is divided into four stages: the prophase, metaphase, anaphase, telophase, and then the cytokinesis. First will be the prophase. In prophase, chromatid condenses, becoming visible. The nucleus membrane dissolves, and can be seen here. This is where the nucleus membrane and the centriole appears and migrate to opposite sides. These are the centrioles that migrate to opposite sides. Then the spindle fibers start to form between them. This is where the spindle fibers here. Then the metaphase. Chromosomes line up on the metaphase plate. Centromeres are attached to spindle fibers. The centromeres of the chromosomes here is being attached to spindle fibers, and the chromosomes is being aligned or line up in the metaphase plate here. Then the anaphase. Spindle fibers contract, centromeres divide, the sister chromatids are pulled away from each other towards the poles. These are the centromeres that are being divided, and then they are being pulled away from each other towards each poles. Here they are being pulled in these poles and poles. Then the telophase. Chromosomes reach poles, nuclear membranes form around the two new nuclei. And in the photo, that is the product of the telophase. The cytokinesis. The cytoplasm distributed equally between new cells. And these are the products. The two new daughter cells that are identical to each other and to the mother cell. Then, type of or kind of M phase, the meiosis 1. Cell division that reduces the chromosomes number by 1/2. It is consists of four phases: the prophase 1, metaphase 1, anaphase 1, and telophase. Start with prophase 1. Longest and the complex phase, or about 90% of the process. It includes the synapses, or or the homologous chromosomes come together to form a tetrad. And tetrad, it is two chromosomes of four chromatids. These are the two homologous chromosomes that are being, or they come together to form a tetrad. This is a tetrad. Then the crossing over. Crossing over, or the variation, may occur between non-sister chromatids and the chiasma. Chiasma are the site of crossing over. Crossing over, segments of non-sister chromatids break and reattach to the other chromatid. These are the non-sister chromatids. After they come together, there is an happening where the break and reattach of the other chromatids in the chiasma, and then the end product will be this, or the recombinant chromatids, and this is the end product of the prophase. The metaphase 1, the shortest phase. Tetrads align on the metaphase plate. These are the tetrads that being aligned in the metaphase plate. Anaphase. Homologous chromosomes separate and move towards the plate. Sister chromatids remain attached to their centromeres. These are the homologous chromosomes that are being pulled towards their poles here. Then the telophase. Each pole now has haploid set of chromosome. Then cytokinesis occurs and two haploid daughter cells are formed. This is in the photo provided, that is the telophase 1. Then the meiosis 2. Separate chromosomes into two chromatids. It consists of four phases: the prophase 2, metaphase 2, anaphase 2, and telophase 2. Prophase 1. Chromosomes begin to condense. Nuclear membrane dissolves. Spindle fibers form. In the photo provided, that is the prophase 1. Prophase 2. Then the metaphase 2. Spindle fibers attach to the chromosomes. Chromosomes line up in the center of the cell. These are the chromosomes here that are being aligned in the metaphase plate or in the equator. Then anaphase 2. Centromeres divide and sister chromatids move to opposite ends of cell as spindle fibers shorten. Here, the spindle fibers contracts, that makes the sister chromatids being pulled toward the poles. Then the telophase 2. Chromosome reach opposite ends. Nuclear membrane forms. Then in the photo provided, that is the telophase 2. Then the cytokinesis, or the cell division. And please take note that the event of meiosis 2 are analogous to those mitotic division or in the mitosis, although the number of chromosomes involved has being halved. In the product of the meiosis is 4 unique new daughter cells can be seen in the photo provided. And that would be